We'll move on now to our next major speaker, which is Professor David Sinclair, who will talk on aging. Can it be slowed or reversed? David is a professor at Harvard Medical School in the Department of Genetics and also here at the University of New South Wales, and an honorary professor at Sydney University. At Harvard, he was the founder and remains the co-director of the Paul F. Glenn Laboratories for Biological Mechanism of Aging, and he's a senior scholar at the Ellison Medical Foundation. After doing a Ph.D. in Australia, he worked in MIT, where he discovered the cause of aging in yeast controlled by genes called the sirtuins that we've heard from Steve earlier. And in 1999, he moved to Harvard Medical School, where his lab works primarily on understanding the role of sirtuins in diseases and aging in mammals. He's also contributed to the understanding of how sirtuins are modulated by endogenous molecules and pharmacological agents such as resveratrol. I got that right, I think. He has been awarded multiple honours for his outstanding academic work, and in 2014, he was in the Time 100 honoree of the Times magazine's most influential people in the world. So that's quite an extraordinary achievement. And like Steve, he's been a very major advocate of the role of science and evidence-based work, and been a public speaker. And last year, he gave a Sydney talk on his topic, and I noticed yesterday that there's over 10,000 views of this lecture on YouTube. He's a great communicator and an advocate of science in this area, so without an on, I welcome him.
Thank you, Professor Farrell, and thank you, Dean Smith, for inviting me to give this talk. It is a great honor, and thank you all for coming. I know you all have lives that you need to get on with, and I appreciate you taking a few hours out of your day to come hear Steve and myself, though Steve is a hard act to follow, it, as you can tell. So tonight I want to talk about a fairly new topic that's I haven't really talked about much, but it's an emerging area in the field of aging research, and something that I think is going to become even more important from this year forward. And I'm going to make a few predictions about the future, so you can look at this video 10 years from now and either be impressed or laugh at what I've said. But yeah, I think the only way to make scientific progress is to try to predict things. That's what we call hypotheses, and I'm going to tell you about a really interesting hypothesis tonight. And it relates to the ability, potentially, not only to delaying aging, as Professor Simpson talked about, but actually reversing aging. And we really do have the emerging technologies to be able to do this now. I do want to give a big shout out before I get going to my lab here at UNSW and Lindsay Wu, the star leader of the lab, and he's a very brave individual taking on this role, and he's done phenomenally well. So thanks, Lindsay, and thanks to the team. We'll be a group of 10 people next year, so we're very excited to be pushing the boundaries of research here at UNSW, which I can honestly tell you, the type of work we do here and the facilities and equipment that we have here in many, many ways exceed that and those that we have at Harvard Medical School. It's one of the reasons that I'm very proud to be here and have moved my research increasingly so down under. All right, so I don't know if you know how long you're going to live, but most of you will only live 30,000 days. That might seem like a lot, but those of you who are beyond middle age know that that goes by incredibly quickly. Way too quickly. And not only that, what's scary about it is that the last 5,000 or so days are typically spent in poor health, declining health, sometimes in pain, and certainly an increasing burden both on the family members and society. So the research that I'm going to tell you about tonight, and the idea is that perhaps we could live 30,000 days, but in good health, perhaps up until the last few weeks of life. And if we can do that, I think we can push the boundaries even further and live another 10,000 days. Imagine being able to live and look forward to living to 100 and playing tennis with your great-grandkids and having 5 or 6 generations around the table talking about all the wisdom that you've accumulated throughout your life. Because I think every time somebody dies, we lose a great deal of wisdom, knowledge, and someone we love. So Professor Simpson set the stage very well. I couldn't have said it better, that our current approach to addressing one disease at a time has worked very well until now. But we're starting to see that we've become very good at treating some diseases like cardiovascular disease, but very poor at treating others such as Alzheimer's disease. And we're ending up a world, certainly a nation, of people who tend to live a long time but don't remember where they are. And this is not the way that we want to practice medicine. It really isn't. So that's why I think the approach should be to keep the whole body healthy and young, not just parts of it. And to do that, you need to study aging. So my point tonight is that it doesn't have to be this way. And I think when it happens, and it might be 5, 10, certainly less than 50 years from now, we'll look back at today's medicine, and think, why didn't they work harder on this topic? Because their world, the future world of our children and their children, is going to be so different than what we currently take for granted and find acceptable today as a normal average lifespan. And I can tell you that there's data on this as well. Here you can see a study that was out of Britain, but it applies to Australia as well, that even though we have longer lives, the percent of our lives that we spend in poor health is increasing, which is not helpful. It's not what we do medicine for. It is to keep people healthier. And so we're going in the wrong direction. I want to reverse this with the research that I'll tell you about tonight. And I'm going to tell a bit of a story. And that story focuses on this person here, who is my mother, Diana. And I use her as an illustration to remind you all that we all have very dear family members that we can help with this medicine. And it's not just about society, it's not just about research. It really is important. And we all have people who we love, who would love to be around for longer and have our children get to know them. So my mum is fairly typical. She's an Australian. She was born in Strathfield. Here she is. It's a little girl, a pretty cute one at the time. And so she ended up being a medical technologist. This is how I remember my mum when I was a kid growing up in St Ives here in Sydney. And she was fortunate to marry my father, and they have been married for 49 years. Now, tragically, 20 years ago, almost to the day, my mother developed lung cancer, illustrated here. And this was just as I was finishing my Ph.D. at the University of New South Wales, trying to figure out what am I going to do with my life. And I wasn't sure. I thought I would study, maybe the sex life of an anemone or something like that. But when your mother is dying, and in this case was given less than 1 year to live, it tends to focus you pretty well. And I decided to devote myself not only to medical research, but to translational research and finding medicines that will help people like my mother not only prevent cancer, but to beat this disease. And remember, this is 20 years ago, and what I was doing 20 years ago was studying yeast cells. Here's one of those yeast cells stained with a yellow dye. This is an old yeast cell. It's true, yeast get old. They live about 20 divisions, and then they die, and they're fairly typical. They get fat and big and sterile. So it's very typical. And I wanted to mention my mentor and supervisor here at UNSW, who's now retired, Professor Ian Dawes, because he was the one who said, David, you've got to go and make a name for yourself. If you want to have an impact, don't worry about your mother. She'll either live or she'll die. It's nothing you can do about it, but you have to go make your mark. And he can encourage me to go to this place, the Massachusetts Institute of Technology. And I also want to mention a person in this audience, my uncle Barry Webb, who's in the second row here. He told me that this was the place to go. He had studied at MIT and thought this was a place where you really can make your mark, and he was right. I'd never heard of MIT at the time, but he was right. This was a great place to go. And actually, you might recognize the top building. This is their main campus, but there's also an auditorium on the bottom here called Kresge Auditorium, which is actually the reason we have an opera house that looks like it does. Sarin and one of the judges in the competition decided that Utsen's design was actually possible based on this auditorium that had just been completed by he and his colleagues over at MIT. Okay, so I headed to MIT as a young boy, 24 years old, thinking that I probably would never see my mother again. Remember, she only had a year to live. But I thought, maybe I can help my children. And so this is Leonard Guarente, my mentor at MIT, and we decided to study, while I was working under his direction, to study aging in those yeast cells and understand, why don't they live forever? Why don't these little yeast cells live forever? Pretty simple cell, should be easy enough. And so what we found out after a few years of intense research was that their aging is due to their chromosomes getting tangled up. And what was causing that was that there were genes that were being switched on and off in the wrong way. Okay, so I'm going to just illustrate that a little bit with a graphic here. So we're going to zoom inside someone's head, because we all have genes. This is what's inside your head. We zoom into the nucleus, and within the nucleus, of course, are the chromosomes. Okay, and you all know that chromosomes, if you untangle them, have DNA, and that's this gray loop here. Okay, so you may not know that the DNA is held together and wound up in protein bundles called histones. So these little balls are called histone proteins. And they're very important, not just because they compact the DNA, but they largely control which genes are switched on and off. So when you eat a meal, these histones will change which genes get turned on, and then they'll close up again and switch a gene off after you've finished the meal. And what we found in yeast was that the reason that these yeast cells get old and sterile and fat and slow down is that the genes that are supposed to be on when the yeast cell's young get switched off, and vice versa. So the pattern of gene expression, it's called, gets disturbed as the cells get older. And what we're starting to realize is that that's probably true for ourselves. And it's a brand new area for science known as epigenetics. Not genetics, but epi, the control of the genetics. And so what I've got here, that on these histones, you're probably wondering, what are those little flashy LEDs? Those are chemical changes on the histone proteins, so they can be acetyl groups or methyl groups. And these chemical groups are added by other proteins or subtracted to change which genes are on and off. So you want to have a liver gene on in your liver, you don't want it on in the brain, for example. And that's how the cell controls it, with these LED chemical groups. And one of the genes that seemed to slow down aging, or did slow down aging in the yeast cells, and now we know slows aging in worms and even mice, is a gene called SIRT1, one of these sirtuin genes that Professor Simpson mentioned. And its job is to control these chemical groups and control which genes are switched off and on when we're young. And what we think goes wrong is that the activity of this protein, this gene called SIRT1, goes awry. Now, it's not the only important gene. Professor Simpson mentioned others, but we think it's a critical component of the system to control which genes are on, which are off. And just to illustrate that again, this is an idea for a cell. So this is a nucleus, and the chromosomes, or the DNA, I should say, these little black strings. And I'm showing you a little red dot is a gene that's switched on. So it's making what's called messenger RNA, so that's a gene that's on. And if you look over to an old cell, it's now switched off. It's shut down. And vice versa, you see a gene that's off might be switched on during aging. And that happens. We know that's a fact. If I took your brain when you were young and old, I could see there's a big difference between which genes are on and off. We think that is a major cause of aging. But that's good news. I know it sounds horrible, but the good news is that that process is reversible. Unlike mutations, which were thought to be the major cause of aging, it's very, very difficult to fix mutations. We barely have any technology to do that. But to reverse this, we know how to do that, and we're getting the tools to be able to do it. And even Time magazine has been excited about this, that your DNA isn't necessarily your final destiny. It's your epigenetics that we can alter and perhaps reset the clock. So we did an experiment. I'm going to share with you data that we haven't yet published, but I think it's so interesting that I can't hold it back. So what did we decide to do? We decided to test this idea. If we start changing the gene pattern early in a young mouse, and we have ways to do that, which we can talk about later. We have ways to accelerate those changes in the genes and make a gene that's meant to be off, on, and vice versa. And one of the interesting things, the way we did that was we took a gene from a slime mold, it's called PPO1, and we know that this enzyme is able to stimulate these changes in the mouse. So we have a mouse slime mold hybrid, okay? I know it sounds horrible, but it's not painful. But we can now ask the question, if you do this, what happens? And when I took a survey of my lab members and my colleagues, they said, probably nothing will happen. And then probably I would say the other half of people said, oh, the mice will die from cancer because you're messing with these systems. Well, that's not what happened. They didn't die and they didn't get cancer. And I'm going to show you what happened. I'm going to show you mice of the same age, except one's had its gene patterns switched on and off earlier than normal. Hopefully I can get this little movie. I took this before I came, so this is hot off the presses. Actually, it's not even on the presses, I should say. Okay, let's try to get this going. Is it going? There we go. All right, so this is one of the normal mice. This one's currently 18 months old. Okay, can you tell? This is one of the ones where we've induced these epigenetic changes. And it looks, to all intents and purposes, like an old mouse. Let me show you a close-up picture. Okay, these mice are the same age. And in fact, they have the same DNA. They have the same genes. It's not their DNA, it's their epigenome that's changing. And we think that's what's causing aging in large part. And it's these genes that Professor Simpson talked about that can actually delay these changes from happening. Okay? And we're at the point now where we're trying to reverse this, because we now think we have a good idea how to do that. But one of the things, well, let me just briefly tell you, these mice, they develop osteoporosis. Well, you can see they're obviously losing their hair and getting gray. That's obvious. But they also lose their memory. They get osteoporosis. They get loss of cardiac function. Their skin looks old. I don't know if you can see the ears even start to look wrinkled. Cells accumulate in the ears and in the skin like an old mouse or an old person, called senescent cells. So we think this is good for two reasons. One is that we now have a system where we can test drugs to slow down aging very rapidly. We can do an aging experiment now in about 1/3 the time. And the other good news is that we think that we may have uncovered a major cause of natural aging that's reversible. So let me put that in a different perspective. The genes are like the recipe book, okay? And you follow that when you're young to the letter, okay? So when we're young, we're like a master chef. We make it perfectly the same every time. This is what it means to be youthful. When we get older, we're more like this. Unfortunately, right? So what we need is a drug to fire the Swedish chef and bring back the master sushi chef. And that's what we're trying to do. Now, I want to finish in the latter part of the talk about the development of drugs that may actually achieve this for the elderly, because it's very difficult even for someone like myself to go on calorie restriction or restrict protein. I don't think I'll order the steak tonight. Thanks a lot, Steve. And chips are out. But this is, of course, very difficult, and it's unwarranted to expect an old person to go on a treadmill or eat a very, very restricted diet. So we need drugs. I think you'll agree with me on that. So it was mentioned by Professor Farrell, this molecule resveratrol. It became famous for its existence in red wine and association, apparently, with good health in Europe. We discovered it not through any of those studies, but independently we did a biochemical screen for any molecule that could remove those chemical groups that I showed you, those little red LEDs. And this was a molecule that stimulated that process to reset the clock. And we didn't study red wine, that's a mistake, but we did use resveratrol to try to understand what's possible. And what we found was that the mice on resveratrol, partly as Professor Simpson showed you, we were able to delay aspects of aging in the mice, both on a high-fat and a low-fat diet. The mice were seemingly resistant to the effects of obesity. Their cardiovascular systems were very clean. Their livers looked great. when we gave their tissues to pathologists, and there are pathologists who study mice for a living, believe it or not, they said that these mice on resveratrol, even though they were on a high-fat diet that would normally shorten their lifespan, they looked just as healthy or healthier than the mice on a lean diet. So this was the first molecule to widely slow down aspects of aging and protect against the effects of obesity. There are other drugs now, Professor Simpson mentioned. This was a leading one. In fact, if you gave resveratrol to the mice, they could run twice as far on a treadmill. And I'm sure Olympians haven't missed that fact. But it's hard to test for resveratrol. I'm sure you could just say, I had some red wine last night, I promise. So resveratrol was a starting point. And actually, to really tell you my family story, my parents went on resveratrol when I first made this discovery 10 years ago. You know, and it's not a clinical trial, so I'm not saying that it's proof of anything. But my mother has been able to see her grandkids, 3 of them, they're from my family, and 2 from my brother. And, you know, it certainly didn't harm her. And we've gone on from there to develop much more potent molecules that are 1,000 times more effective than resveratrol. This is an example of that SIRT1 enzyme that controls those LEDs. And the pink molecule is one of the drugs that's in development at GlaxoSmithKline. GlaxoSmithKline is pouring hundreds of millions of dollars into this field, trying to make a drug that could truly slow down aging, if not a broad set of diseases. And you might ask, well, what if you feed these drugs to the mice? Well, in collaboration with Rafa DeCarbo at NIH, the answer is, well, it does pretty good things to the mice. They live longer. 2 of the experimental drugs shown in the blue and the green. The mice have less osteoporosis. Their muscle strength is better. They're not prone to muscle wasting. Their blood glucose is better. They run twice as far on a treadmill, just like resveratrol. So this is good stuff. And actually, the one called 2104, shown here in blue, has been in humans, and it does appear to be safe, at least in early clinical trials for psoriasis, and possibly even working for that inflammatory disease, that skin disorder. And so next year, more trials will begin with even better molecules than this. And so just to get back to the personal side, this is my mom doing really well on resveratrol. And things were going great, as Professor Farrell mentioned. I even made Time magazine, and oh, this is not me, by the way, if you're wondering. I was in the back somewhere of this magazine. And you know, of course, when things are going great, that's when you really have to be thankful. Because just as this was happening, a few months later, I received an email from my dad just to say, Mum's become sick. And of course, he was very kind not to make me panic, but this was very serious, and I rushed down to Australia. You can see this is fairly recent, it's just in September. And fortunately, I was able to see my mum. She was in hospital and actually recovered pretty well. This is one of the last things that I was able to take of her. In fact, I accidentally hit the record button and inadvertently recorded the last movie. It was only about a couple of hours after this that she passed away, quite unexpectedly for us because she was doing so well. But there's nothing more than this sort of thing in a family to make you want to work harder. And I've actually said to my dad, and remember this on my gravestone, can you please print, if only I'd worked harder. But it's serious stuff because it will really impact lives. And I just want to reiterate that we're currently only given 30,000 days, and I don't think that's enough. I don't think we spend enough of that time in health. And it really doesn't have to be this way. And looking forward, we have new technology that's even better than I've told you about. We have a molecule called NAD that turns on a whole family of protective enzymes called the sirtuins. There are 7 of these. We've got molecules that can raise the NAD levels back up from old to young again. The way we do it is we give a molecule called NMN, which is a precursor molecule to NAD. So if you feed this or inject this into mice, the low levels of NAD, when they're old, go back up to young levels. And in humans, what happens as we get older, our NAD levels fall down, and our ability to protect ourselves goes down as well. And so what we found was when we gave mice this NAD boosting molecule, aspects of their aging were reversed in just 1 week. We looked at their muscle, and so Lindsay Wu, who's here, UNSW, helped us with that study, and Nigel Turner here, also from UNSW, we were able to show that, at least for muscle, it's very easy to reset aging. It only took a week, as long as you know what to give. And I think this is the beginning of a new era in aging research where we can not just delay aging but reverse it. And people catching on, reversing aging, it's not as crazy as you might think. The lab at UNSW is pressing forward. We've been not criticized but challenged because this all sounds too good to be true. Maybe NMN and NAD will cause cancer. Well, here's research from Lindsay's work. If you feed mice that develop liver cancer, on the left you can see these little dots here. These are tumors that will kill a human pretty quickly. These are liver cancers. And if you just put NMN in the drinking water of these mice for a few months, you barely see any tumors. So this is not a tumor-promoting molecule, at least in liver cancer. So we're excited about this research, not just from an aging perspective, but ability to really prevent and treat major diseases as well. And lastly, this is my son, Ben. He's the youngest in the family, a budding scientist. Here he's doing a genetic test. Fortunately, he doesn't carry the mutation that led to my mother's lung cancer. I do, but he didn't get it, thankfully. But I want to use him as an example of the bright future that the next generation and his future descendants will have. We have new technology not just to reverse aging, but to go in and correct genetic diseases, to improve fertility, to sequence our genomes and know how to live and what to eat. And I think one day, maybe it's for our generation, maybe it's for his generation, but there will be a day when we will have 6 generations around at the dinner table talking about the good old days. And when that happens, I can't imagine anything better than that. Thank you very much for coming.